small groups, 1 or 2 of you by yourself, 1, 2, or 3 of you. Pick one of these 4 sequences that one of you submitted. These are real submissions from you. 4 different ones. Pick one, double check. Does it meet the requirements of the exercise, which were, just a second, what were they? 15% A, and what else? 35% G. Okay. So we take about 5 minutes to do this. We'll come back together. I'd like every group or some of your groups to report back which one you looked at, 1, 2, 3, or 4, and whether or not it's right or not. Okay, go ahead. Good question up front here. So what was this assignment to generate?
A
single strand.
Including 15% adenine and 35% guanine.
Okay, so 15% adenine, 35% guanine, as I wrote up there, not guano, that's something else. Up in the upper right-hand corner. So these are all single-stranded sequence submissions. So what's the problem?
They're not complete.
So which one are you working on?
Oh.
Which number?
No, I just realized that I did wrong the homework. That's what I was asking.
Okay, so does anybody else have any? Yeah, please.
So just to clarify the confusion, what I did was now I understand it said single strand, but I took in DNA, which was usually 2 strands. So what I did was, in that case, does that mean that I created 2 separate strands with the same percentage differences? Because C and T and
every time you write a T on one strand,
C and A and all that,
there's an A on the other strand.
Right.
That's Chargaff's rule. So if we wrote out the full double-stranded, look, number 2, who worked on number 2? It was the easiest one, right? It's the shortest one. Just one of you. Oh, come on. So does sequence number 2 match the requirements for this exercise? Does it represent a double-stranded molecule of DNA that is 15% A?
Actually, it's, based on complementary.
Let's write in the complementary strand.
We did, right?
But then there's that rule, though, that there has to be that 1-to-1 ratio. Wouldn't it mean that it's on that strand as well, then?
So there's the double-stranded sequence.
And then you have 15, 15, 35, 35.
How many total nucleotides?
Right, so you need 3 adenines and 7 guanines.
That's 20 nucleotides.
And you need, like you said, 3 thymines, 3 adenines.
Yep.
Make a 30%, and then 7 cytosines and 7 guanines to make 7.
So 3 out of the 20 are
50%.
A, 15%.
Yeah.
So that one gets a check. That's right.
I think my question was, well, because we had gone over Chargaff, what I had applied to was I just took 12 instead of 20, and I rounded up or rounded down. Because he did that.
That's okay. Yeah, that's okay. Yes, another question. Please pay attention because these might be useful answers that I'm going to be giving to questions that you might be having. If you're not having class-related discussions, please exit.
For number 2, which we just did, I pretty much wrote both strands rather than just...
That's fine. Yeah, so if you wrote both strands, and again, the reason that we're going through this in class is because this is a good way for you to get feedback. I didn't get a chance, of course, to show everybody's answers up here. We're just doing some examples, but hopefully you'll ask me questions now so that you can figure out for yourselves whether or not what you submitted was accurate or not. So yes, if you wrote out the full double strand, as long as it met Chargaff's rule, equal number of A's and T's because of base pairing, and as long as the percentages are correct, that's fine. Okay. Yes, ma'am.
So if we put one strand, if we should be correct?
Yes, exactly. So one strand was fine. So here was a strand, and I was glad to see this one because it doesn't contain any A's or G's, right? At first glance, you might look at that and say, that doesn't even come close to approximating what I was asking for because there are no A's and no G's. But you have to realize, and based on the video you watched, that when we write a single strand of DNA, we just don't write both strands of sequence normally because you can figure out the second strand of the DNA sequence by base pairing. So normally when we write sequences in class, it'll just be one strand. Yes?
So for the homework assignment, would it have been acceptable to write something like number 3 with T's and
Absolutely. So did anybody actually go through that one and check? Was it okay? Yeah, so I'm seeing heads nod. I checked number 3, it's fine. So how many nucleotides are in number 3? Let's go
20.
through this one more example. So we've got 20 nucleotides.
3 T's, 3 A's, 7 G's, 7 C's.
7 G's, 7 C's.
I did that one. So.
Okay, there you go. So we've got 20 total nucleotides, 3 of which are A, 15%. If we wrote in the second strand, what would happen? We would have how many nucleotides?
40.
We'd have 40, and that would mean there would be how many A's?
6.
6. Because for the 3 T's, they're paired with A's, and the 3 A's are paired with T's. So then that would be 6 out of 40. It's still 15%, same ratio. So all of these, 2, 3, and 4 were accurate. Whoever submitted those were all correct. And I actually think I created number 1, which is the only one up here that's not quite right. I think I miscounted. So, oops. I was close. It was something like 16.4% A instead of. Yeah, right. I never give, I give recognition when you catch errors I make. I don't necessarily give extra credit when you catch errors I make, but I appreciate it when you check my work.
So I have a question. So like for the exam or something like that,
Yeah.
would your preference be for us to write it out like number 2, or is it okay if we do it like number 3, or is there like
This is,
method you prefer?
I'm glad you're asking. I will be very specific when I write exam questions. I'll try to make the format that I expect as clear as possible, but I bristle a little bit. I'm kind of actually getting a little bit of goosebumpy because I'm trying to, again, help everybody understand that it's okay to be creative in a science class, and I'm happy to be, and I hope Fatima is happy to be, flexible as well, grading. Because as long as you demonstrate that you understand the material, that's the important part. Not quite as much the formatting. But yes, I will try to be as clear as possible to reduce stress and question asking during exams.
I wish my chem teacher was like that.
Well, organic chemistry is a totally different beast where, well, I don't know, but yes. Yeah, you have to know which sort of molecular structure you're doing, which sort of representation of the molecules.
So for example, by saying like in a research problem, if you already know the percentage of one of the DNA strands, then you already know the percentage of the whole DNA.
That's a fantastic point. Did everybody in the back hear that? I could give you, and I'm sorry that you said that out loud, and maybe if you didn't hear it, close your ears, because I don't want you to hear the rest of this. That's one of my favorite exam questions.
Oh, okay.
So let's do it. If I tell you that an organism's genome is 17% adenine,
You automatically know.
what percent guanine? Okay, so if there are 17% A's, what's the percentage of T's? So it would be 17% T.
33%. 33% G.
Okay,
3% C.
so that should add up to 100%, hopefully. So now you've got an edge up on, a leg up on the exam. Yeah, exactly. That's one of the nice things about Chargaff's rule. Okay. So here are, what are these? Chromosomes. How many chromosomes are there?
6.
Right, no, so now we get in 6, 12, 24, 48. Let's just keep multiplying by 2. So this is what is this object? What do we call that thing I circled in blue? A chromosome.
Oh, sister chromatids, actually.
Yeah, and
Oh,
we'll,
no, sorry, but
yeah, sister chromatids. Each of these objects is a double helix. There's 2 double helices there. So I'm going to circle 1 double helix in red. So that's an entire chromosome circled in red. That's 1 chromosome. So when we call that X-shaped thing a chromosome, we do that colloquially, and people out in the community do that as well. We look at it, it looks like a single object. It's really 2 molecules, 2 double-stranded double helices that are next to each other. They happen to look like an X. It's not 1 object, it's 2 objects that are crossing each other. So the correct answer to that is 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Now, for those of you that said there are 6 chromosomes, maybe you were right, because there's a little subtlety here. This chromosome spread is from a muntjac. If you don't know what a muntjac is, you can Google it right now because you've got the internet. They're cool-looking animals. One of the reasons that geneticists study muntjacs is because of mammals, they have the fewest number of chromosomes. So it's easy to look at this is their entire genome. So we just decided that we're looking at 12 double helices. How many chromosomes do they have? And this is just supposed to be a little bit of refresher and a little bit of projecting what we're going to be talking about next week and for the next couple of weeks. What's the relationship between those 2 double helices? How are they related to each other? So they're sister chromatids, which means
one came from one parent, the other one came from the other.
how did that organism get, this organism has 2 copies of that chromosome. One of these double helices came from its mom at fertilization, and one of those came from its dad. Wait a second. Is that right? No. These 2 chromosomes that are juxtaposed next to each other that look like an X are the 2 copies of mom's chromosome. This is right before the cell divides. This is metaphase. Well, again, we'll go into more details later. I'm just trying to show you where we're headed in class. So whenever you see a chromosome that there's a picture and it looks like what we call a chromosome, colloquially speaking in common language, that's 2 copies of the same chromosome right before cell division happens. So where's the version of that chromosome that came from the dad? Because all of us have 1 copy of each of our chromosomes that came from our mom at fertilization and 1 that came from our dad. Which numbers?
1, 2.
Yeah, chromosomes 1 and 2 that I drew up here in red are that same chromosome but from dad and a copy. So let's letter these chromosomes then. We'll say that's chromosome A. So you've got chromosome A from mom, you've got chromosome A from dad. Because we're all made up of genetic contributions from both of our parents. This muntjac then has 4 double helices. At this point in time, in this picture, there are 4 double helices, 1, 2, 11, and 12, that are all the same chromosome. They're all chromosome A. So how many different chromosomes does a muntjac have? It's got, right, it's got 12 total double helices up there. 4 of them are chromosome A. 4 of them are chromosome B. I'm calling them A, B, and C arbitrarily. So which ones should we call chromosome B?
3, 4, and 9 and 10.
Okay. So those we'll call chromosome B, and those, which happen to look the same, which is how we know that they're related chromosomes, we'll call chromosome C. So the muntjac has 3 different chromosomes. In humans, how many chromosomes do we have? How many different chromosomes? It's either 23 or 46. We have 23 different chromosomes numbered 1 through 23. I'm just saying here muntjac has 3 different types of chromosomes, lettered A, B, and C. We're going to practice this more as we go through class. It's a strange concept, but it's an important one. Any questions or concerns at this point? Time for a question or so.
So the point of this exercise was that there is 12 strands, but there's 3 different chromosomes?
12 double helices. So, since you said the word strands, each double helix is made up of how many strands? 2. Okay, I just want to make sure that, because this is where you might get lost, where I definitely got lost back in the day, was all of this nomenclature. What makes up what makes up what makes up what? So 2 strands per double helix, 2 double helices here per 2 chromosomes. Right, that's 2 double helices. So a double helix is a chromosome. 1 chromosome, 2 strands. So the point of this exercise really is to start talking about nomenclature of chromosomes. Strands, chromosomes, and then the different chromosomes, here A, B, and C, for example. Good. Now officially today we're supposed to be talking about prokaryotic chromosome structure. So this is eukaryotic, by the way, that was just an example. All we need to do prokaryotic, what are the common features of prokaryotic chromosomes? What do you know about bacterial chromosomes?
They're not enclosed in a nucleus.
There's no nucleus. That's why they're prokaryotes.
Also circular.
And circular. That's the key thing I want everybody to remember about bacterial chromosomes, prokaryotic. They're circles, as shown here. So you can see a cartoon of a bacterial chromosome which has no ends. It's one big loop. And if you don't believe me, then you can look at a transmitting electron micrograph of a bacterial chromosome, and you can see that in reality it is a loop. It's not just how we draw it. Bacteria, the only other thing that I want to mention briefly about prokaryotic chromosomes, their big chromosome there that's shown as that big massive spaghetti on the left is a circle. What other DNA do prokaryotes have that's shown up there? Plasmids. So what are plasmids? What's the value of a plasmid? What do they do? They're DNA, just like the big chromosomal genome, that big loop up here. So plasmids are used.
So it's either DNA that's going to be transferred or maybe was accepted from another cell?
There's these little packets of DNA. They're round, they're circular still, but these are DNA that get moved from bacterium to bacterium when they have sex. You didn't know we were going to talk about sex and genetic. Well, you probably should have assumed. Because at some point we're going to look at pedigrees, and then we have to talk about sex. Did you know bacteria have sex?
Yes.
Okay, good. Well, at least the front third of the class. Back 2/3 of the class, did you know bacteria had sex? What's it called when bacteria reproduce?
Conjugation.
Well, it's conjugation. It's not technically sex.
Multiple different ways of interacting with each other sexually.
But it's how... Let me take a step back. Literally. What's the purpose of sex?
To reproduce.
Boring. Oh.
No.
Fun. Sure. Right. to exchange genetic material, to pass on your genes to the next generation, to reproduce. To a geneticist, and I'm not saying you have to claim to be a geneticist at this point, but to a geneticist, sex is new combinations of genetic material coming together to produce different, genetically different offspring. That's not quite what bacterial conjugation is, but in bacterial conjugation, two bacteria come physically close to each other like a man and a woman do, and the birds and the bees and all that, but they connect to each other. What's it called? A pilus?
Yes.
A pilus. They form a pilus, a bridge that spans the two cells, and DNA can go back and forth through that temporary bridge bond between the two bacteria. And it's plasmids that get shuffled back and forth, and that's how bacteria share DNA. How is this relevant to us all? Current genetics.
About bacteria or about like?
Yeah, about why is it important that bacteria can have sex?
Or
Why is it important that they can share genetic material with each
So
other?
that they can become resistant to like medication,
Ah,
antibiotics.
antibiotic resistance. So when bacteria share plasmids with each other, most often what gets transferred are plasmids that carry genes that help them survive being bombarded by antibiotics. When we use antimicrobial hand soap and all those other products. So this is really important because this is how bacteria become resistant to, multi-drug resistant to lots of different antibiotics. If they have a gene, what does a gene do? I should have asked that on the first day of class, right? What the hell are we doing in genetics? We talk about genes. Why do we care about genes?
Because they express themselves either like physically or.
So what is DNA and what is a gene?
It's a recipe for a protein.
Recipe for a protein. So genes get expressed, it's a term we use, means that genes get read by some machinery in the cell and produce a protein. Why do we care about proteins?
They're the structure.
They're what you see before you, other than my clothes. Right? Everything we're made up of is protein, almost. And we would be dead if we didn't have proteins because, I don't know, name a protein. Hemoglobin, great. Okay, I would be dead if I did not have hemoglobin. I would asphyxiate pretty quickly, right? Because hemoglobin is a protein that binds oxygen and delivers it to our tissues. Name another protein. Myoglobin, that's GA. Keratin. I wouldn't be able to bite my nails if I didn't have keratin. Which one? Right, so that's a great example. It's a class of proteins, enzymes, the ones that actually do biochemical functions in our cells. Like the ones that make our cellular energy, ATP, that we burn to do the work of the cell. So that's why we care about DNA, because it contains genes, and that's why we care about genes, because they produce our proteins and make us alive. So that makes genetics pretty cool. It's the study of life. And it's very useful for these bacteria to have genes because they help them evade things that we're trying to use, like antibiotics, to kill them, and things that other bacteria produce, like antibiotics, that they try to kill their other bacterial friends.
do they also have RNA?
Do bacteria have RNA? So any organism that has DNA will transcribe it to produce RNA that is then read to produce proteins, which we talk about in the central third or the last half of class. Your most beloved topic, transcription and translation.
So through the genetic sharing of two different types of bacteria, we can have new species of, or
Oh man, so then the question is about species, which I'm going to defer until later, but it's a very interesting question. So briefly, yes, but we'd have to have a long debate, which I'm not willing to entertain right now, about how you define a bacterial species. It's complicated.
So along those lines, during conjugation, you're not necessarily producing a new bacterial cell yet, right? They're just passing the information back and forth.
Correct.
It isn't until they...
Cell division, then you've got two offspring daughter cells that are genetically different. Yeah. Okay. So I wanted to touch on one more point that was brought up last class. It was an excellent point. Let me pull up another image here. So we need to talk about hydrogen bonds, just briefly. So there are your nucleotides. Organic chemistry students, which all of you have been or should be or are now. What are the solid lines that connect all of the different atoms? Those are chemical bonds. So there's a double bond, for example, between two carbons. What kind of chemical bond is that? Covalent. And then everybody goes narcoleptic and falls asleep. Okay. Not that I make fun of the narcoleptic, because that's a serious disorder. We'll learn more about that in the semester, potentially. What are the dashed lines? Hydrogen bonds, right? So this represents two different types of hydrogen bonds. That's all I need to say about this. Other than, which is a stronger bond? Hybrid, pardon me there. Which is the stronger bond? Covalent bonds are stronger. It takes more energy to break them than hydrogen bonds. Hydrogen bonds are pretty weak. What's weaker than a hydrogen bond? Van der Waals interactions. Sorry, I was a biochemistry major, so I like to put, I know, this is genetics class. So in order, covalent bond is stronger, hydrogen bond is weaker. What's the easiest thing we can do to break hydrogen bonds? Eat? Did you bring enough to share?
Add water.
No, did you say eat or heat? Heat. Okay. I know, we're all hungry up here. We all heard eat. Okay, so from left to right, and from right to left, this is what happens when you change to the temperature to a double helix, right? As you heat, those hydrogen bonds that bind the two strands of DNA together do something. What do? They break the bonds. What other words do we use for that? Denature. And then colloquially, what happens when you get really hot? You melt. So often geneticists, and we'll talk about this later in the semester as well, we call this melting. You heat up a piece of DNA, it melts, it denatures, it unwinds. You break all the hydrogen bonds. And then as you cool it down, the two strands, not denature, but renature. Those hydrogen bonds form again. If you were a cell, and we were trying to do what the cells do, which is to read their genes, produce proteins so that the cell can survive, the organism can survive, we have to inquire somehow about the actual DNA sequence, right? That's what encodes the protein. It's the sequence of nucleotides, A, T, G, C, T, A, T, whatever. What is it in the cell that's going to read the sequence of your DNA and make RNA that encodes protein that produces enzymes that keep you alive? What reads the DNA?
The little one inside, it kind of opens and closes.
So right, so there are lots of enzymes. General answer. It's proteins that read our DNA. It's kind of chicken and the egg. So we have proteins that read our DNA to produce proteins, that read our DNA to make more proteins, that we use to synthesize new DNA that then we need to read to produce more proteins. So if you're a protein, how are you going to read the sequence of this double helix on the left? So what's the information? How can a protein, just attaching itself to a molecule of DNA, how can the protein figure out what the letters, the nucleotides are that it's touching? Number of hydrogen bonds between the base pairs? Okay, so C's and G's have 3 hydrogen bonds, and A's and T's make 2 hydrogen bonds. But
Are we talking about codons initially?
that's not necessarily, right? So here, I want to point out this drawing of DNA. There's a major groove where the 2 backbones, the 2 strands, the carbon phosphate backbones are farther apart. They make a wider groove in the DNA. And then there is a minor groove that's a bit narrower. You might have heard about this before. Helpful because if you have this sequence, and that means something special, let's say that's the 5' end and the 3' end. Here's the double strand with its polarity. 3 hydrogen bonds, 3 hydrogen bonds, 2 hydrogen bonds, 2 hydrogen bonds. Let's say it's the CGAT that encodes, I know 4 nucleotides does not a protein make, makes 1 amino acid plus 1 more nucleotide, but let's say that's the important strand. If an enzyme is just reading, if it's sitting here, for example, let's say that's an enzyme. It says, okay, I'm attached to a nucleotide that can form 3 hydrogen bonds. How is that helpful? Which nucleotide is it that can make 3 hydrogen bonds? C or G. So proteins, do they want to use the number of hydrogen bonds to figure out what nucleotide they're interacting with? No, because it's ambiguous. If the protein, what else would a protein have to do, and then you, what else would a protein have to do to actually assess how many hydrogen bonds are being formed between 2 nucleotides?
No, the nucleotides.
It would have to,
No, the nucleotides.
it would have to separate the 2 strands of DNA and stick itself in between the 2 strands to assess, to feel chemically, how many hydrogen bond base pairing possibilities are there in between these 2 strands. Enzymes don't usually do that. Some do, but most don't actually separate the 2 strands. There's something else that enzymes can do to inquire about the sequence of the DNA that they're touching.
Well, don't enzymes fit like a lock and key, they say, so maybe they would have a different shape, they have different shapes that would fit into this protein?
Okay, so shape of the enzyme is important.
Right, so whether it has 2 or 1 ring.
Right. So there's other information. Let me give you the answer, and then I'll take a couple other questions if we've got time. So what information does the protein use to inquire about the sequence of the chromosome? We're talking about chromosome structure. Main point here is that most proteins don't melt the chromosome, break the hydrogen bonds to figure out what sequence they're interacting with. So, if you've got a QR code scanner, go for it. Otherwise, grab a web browser and type in this, I know it's not a tiny URL, but short URL. Hopefully everybody's browser will look at this. Tablets, yes. Phones, I don't know. Good luck. It might work, I don't know. I'm prepared to be pleasantly surprised. Okay. Getting success, failure. Yeah. Yeah, you're right, this is going to be a molecular viewer. I'm going to give you a couple more minutes to open this, and then I'm going to open that on my tablet as well. Yes, you can touch it and you can rotate it. Okay.
Yeah, the helix one, and then the nucleoid one, and then, yeah, but it was this, and then got nice little these.
Ah, success. So hopefully everybody's seeing something that looks like this. All right. So, discussion. What are you looking at?
A protein.
It's a protein. So there are two different representations of molecules and atoms up here. So I heard some people up front talking about what we're looking at. So what are these? It's a protein and it's a piece of DNA. So we're looking at, this is a crystal structure. I didn't make it. I found this information online. And then you get the benefits from it. So it's a crystal structure. It's the actual physical structure of a part of a protein bound to a specific sequence, a double helix of DNA. So can you rotate that picture so that you're looking down the center of the double helix?
Yeah, we can.
Right down the middle of the double helix. Yeah, that was good. So something like this. Whoop. There we go. Right down the barrel of the DNA. The double helix is going into and out of the board. So which molecule, how do you know which one's DNA and which one's protein?
The plated sheets in the alpha helices, usually are proteins.
So you might notice, please notice, that there are two different formats of structures here. The DNA, which I'm circling in blue, is being represented by sticks. So each of these sticks is an atom or a bond. It's a bond. So there are atoms at each of the vertices. And we're looking at the bonds. And presume each of those different colors represents a different nucleotide. Then the ribbon diagram is what that's called, circled in red, is a protein. And you can see, as some of you were discussing up here, you can see beta sheets, and you can see helices. And that's not important, that's biochemistry. You don't have to worry about that right now. But now what I'd really like you to do, and the point of doing all of this, is figure out, this is a DNA binding protein. This is a protein, I'm not going to tell you what it is yet, we'll talk about it later in the term. This is a protein that recognizes a specific sequence of DNA and binds there as it's found here. How is this protein contacting the DNA? How would you describe how this protein is inquiring what nucleotides it's associating with? So take a minute, rotate the figure, and discuss how would you describe to me in words how this protein is fitting into or touching the DNA?
It's like she said.
Take a minute, everybody discuss in small groups, and then we'll report out. Okay, so that was a minute. What did you come up with? What's your description? How is this protein fitting into the DNA? How is it touching the DNA?
It's not touching it.
It's not, well, it is, It doesn't look like it, but it is actually physically, well, we can discuss what it means to physically contact something when you're talking about electron clouds, but, sir?
Most of the baryonic or van der Waals type of interaction.
So it's going to be a lot of van der Waals interactions. There is some hydrogen bonding going on. We can't see it in this angle, but are you familiar with the concept of DNA being groovy? Groovy. No, major and minor grooves. DNA has two grooves, the major groove and the minor groove. It's up again. Which groove is this protein interacting with?
The major group.
Is it the major groove or the minor groove? This is actually, it's interacting with the narrower of the two grooves. So this is a minor groove binding protein. It is. Regardless of what it looks like, this protein, I know for a fact, is a minor groove binding protein. This is weird. This is an exception. Most proteins don't interact with the minor groove. Most proteins bind in the major groove, partly because it's bigger, and it fits more proteins better. There's another reason that we're going to get into next time. I just want to leave the last couple of minutes to make a couple of announcements that are important, and we'll come back together for next class. To be continued. So here's the usual what to do for next class period. Watching a movie, we've got a chapter of one of the textbooks to read. I want to add, for those shared Google documents, I got a lot of requests, or invitations rather, for me to collaborate. Thank you for all of you that did that. More than half of you used the wrong email address. I still got the invitation, but it needs to go to my mail.fresnostate.edu address, not my @CSU-Fresno address. Also, please add Fatima, our TA, as well. Please do both of those things, and we'll give you feedback. Hang on, not quite done yet. Thank you. This is going to be the most important thing. If you are not the person that created the Google Doc, make sure that that person, or all of you individually, because you're all collaborators, write your name on the very top line. So we want to have at the top line of the document every group member's name. That's how we know who's in each group. If your name is not there, you are not part of a group. And that equals 0 points. Okay? Then finally, for next class, bring an answer to that question at the bottom of the screen. Okay? Office hours start in 10 minutes.